Welcome to this explainer. Today we're jumping right into a fascinating and, honestly, potentially game-changing new development in neuroscience. Now, if you follow brain health at all, you already know that Alzheimer's disease is one of the most stubborn, complex, and frankly heartbreaking puzzles in modern medicine. Millions of people are affected worldwide, and for decades, scientists have been hunting for one specific mechanism. They've been trying to figure out exactly how this disease operates at the absolute microscopic cellular level. Well, today we're unpacking a major breakthrough that might literally solve this long-standing medical mystery. It gives us a whole new lens to view dementia, and it could completely change the trajectory of future research. Let's get right into it.
So here's the big question. How exactly do toxic proteins kill brain cells? For decades, researchers have known that these toxic proteins pile up in the brains of people with Alzheimer's. You've probably seen the brain scans, right? The literature always talks about these protein buildups, specifically amyloid and tau. They're the usual suspects. But one huge fundamental question remained totally unanswered. How do these proteins actually pull off the crime? We knew what was happening, but the specific how was a total blank. It's kind of like arriving at a crime scene, finding the weapon, but having absolutely no idea how it was used to cause the final damage.
And honestly, that perfectly captures the frustrating reality of this decades-old mystery. We've known the toxic proteins were the executioners, but exactly how they carried out that execution, that's been the missing piece of the puzzle, and it's been driving researchers crazy. It's like knowing a building collapse, but not knowing if it was brought down by a wrecking ball, an explosion, or just a hidden structural failure. And without knowing the exact mechanism of cell death, it's incredibly difficult to intervene. You can't really design a targeted drug if you're just swinging in the dark.
Which brings us to Section 1, Discovering Karyoptosis. Let's look at what scientists just found.
Okay, so scientists have identified a previously completely overlooked process, and it's called Karyoptosis. Basically, this is where the actual nucleus of a brain cell breaks down and collapses. To really wrap your head around how devastating this is, you got to remember that the nucleus is the absolute core. It's the brain of the cell itself. Imagine the central command center of a highly secure facility just imploding on itself. That is the level of horrific physical destruction happening right inside these brain cells. It's a structural failure at the most fundamental level. And the best part? This is the exact mechanism of destruction scientists have been hunting for. The mystery of the how finally has a name.
Moving on to Section 2, the triggering enzyme.
Right, so now that we have a name for this cellular collapse, scientists needed to find the culprit. What actually flips the switch to start karyoptosis? Well, this recent study successfully pinpointed a key enzyme called p38-MAP kinase. And wow, this is a massive revelation. It completely rewrites our understanding of the disease. You see, the toxic proteins don't just magically kill the cell by hanging around it, nope. Instead, they act as messengers. They initiate a process involving this specific enzyme. So the proteins are the warning sign, but p38-MAP kinase, that's the actual executioner pulling the trigger, causing the cell's nucleus to break down.
So let's look at how this builds into a deadly chain reaction. It goes exactly like this. Step 1. The toxic proteins accumulate in the brain. We've known about that part for years. Step 2. This dangerous buildup triggers that p38-MAP kinase enzyme we just talked about. Step 3. Karyoptosis kicks in, meaning the nucleus itself fundamentally collapses. And finally, Step 4. Inevitable brain cell death. It's a super precise, 4-step domino effect. And by finally having a clear map of this progression, researchers now know exactly where they might be able to step in and stop those dominoes from falling.
Which leads us perfectly into Section 3, Hope for Future Treatments.
So, the million-dollar question. What happens if scientists try to interrupt this newly discovered pathway? If we know how the dominoes fall, can we catch them? To figure this out, we can look at a really fascinating comparison from recent lab experiments. On one side, when the pathway remains active, the destruction just keeps going. The nucleus collapses and the cell dies. But on the flip side, scientists found that by deploying a biochemical shield, specifically blocking this p38-MAP kinase pathway, they remarkably delayed brain cell death. Literally by just neutralizing the trigger, that final stage of cellular collapse was stopped in its tracks. The cell survived way longer than it would have otherwise.
And by successfully delaying cell death in the lab, this opens the door to entirely new therapies. I mean, think about it. If we can replicate this blocking effect with targeted medication, we could potentially slow the progression of this devastating disease in humans. This is a massive paradigm shift for neuroscience. For years, all the research focused almost entirely on trying to scrub away the toxic proteins from the brain, which has proven incredibly hard to do. But now we have a way to potentially protect the cells from dying, even if those toxic proteins are already there. We're shifting from cleaning up the threat to building an impenetrable defense for the cell itself. Now, we definitely need to take a step back and add some necessary caution here. While this is an incredibly promising breakthrough for medical research, we've got to remember, it's not a cure yet. The leap from a successful lab experiment to a widely available treatment for humans, that is a long, rigorous road. This is vital science news, for sure, but it is currently strictly in the laboratory phase. The main focus right now is on slowing the progression, understanding the deep mechanics of the brain, and hopefully, eventually, buying precious time for people affected by the disease. So it's about tempering our expectations, while still absolutely celebrating the incredible progress being made.
All right, let's quickly summarize the key takeaways of this incredible news and why it matters so much for the future of brain health. First off, this breakthrough solves a decades-old mystery of exactly how these brain cells die. Second, it identifies the actual mechanism, Karyoptosis, and the exact biochemical trigger, the p38-MAP kinase enzyme. Third, initial lab tests successfully delayed brain cell death by actively blocking that pathway. And fourth, it paves a very clear way for future therapies designed to slow down Alzheimer's progression in a totally novel way.
As researchers involved in these efforts have noted, every scientific breakthrough brings us one step closer to protecting millions of people affected by dementia worldwide. I think that thought beautifully captures the overarching hope of all this neuroscience research. It grounds the hardcore cellular biology back to why we do this in the very first place, to protect the millions of families worldwide facing this condition. Every single time we understand a new enzyme or map out a cellular collapse like karyoptosis, we gain vital ground in the fight. We're turning what was once a total mystery into a solvable puzzle.
So, I'm going to leave you with this provocative thought to ponder. As we rapidly decode these complex cellular mysteries and actually learn to block deadly enzymes in the lab, how close are we to a world where dementia's progression can finally be stopped entirely? It's a profound, awe-inspiring question to consider as we look to the future and track these exciting advancements. Thank you so much for joining me for this explainer. Keep questioning, keep learning, and keep your eye on the incredible, ever-evolving world of medical science.